Hi guys, welcome back to another Khan Academy video. It's been a while. Okay, so today we're going to be talking about 6.2 and D2, digestion and absorption. Um, okay, we're going to go through this as if like we're an apple. Okay, as if we're an apple and we're starting out in the mouth. And you took a bite of this apple and we're seeing what happens to it across the road. Here's a little piece that we ate. Okay, so starting out with the mouth. The mouth is responsible for mechanical and for chemical digestion. So how's it mechanical, you may ask? Well, we use our teeth, which is a voluntary action that we choose to do. It's not automatic. And the chemical way we digest is through the release of enzymes. So what enzymes are produced in the mouth? Well, we have the salivary amylase, which is produced by the salivary glands in the mouth. Okay. The salivary glands in the mouth. And these salivary glands in the mouth function at a pH of around 7. So that is their optimal over here. So you have some glands over here. They're producing the salivary amylase. It's digesting the starch in this little piece of apple. So what happens to our brain when this happens? Well, this food is going to go down the esophagus. It's going to head down, and it's going to go down through using peristalsis. So peristalsis is when the circular and the longitudinal muscles are able to contract and eventually push the food all the way down. So the circular ones are responsible for making sure it can't go backwards. So the food's always heading in a down direction away from the mouth. And the longitudinal muscle is responsible for pushing the food. It's going to pinch it and kind of like get it really go down there. Okay, so once the food enters the stomach, we're going to head into the gastric phase. And this is when distension is going to happen in the stomach. So the food's going to brush up against the stomach from the outside like this. And that's going to send a message up to our brain, actually. So we're going to go back. We're going to go back up there. And we're going to look at the medulla oblongata. So the medulla oblongata. Okay, so what's going to happen here is that when we think about food or when we eat food, the medulla oblongata is going to be like an impulse is going to go to the medulla oblongata, and the medulla oblongata is going to send an impulse through the vagus nerves into the stomach so it can produce, let's say via vagus. And so it's going to be able to produce HCl, hydrochloric acid, and pepsinogen. So HCl is going to be coming from the parietal cells, and the pepsinogen is going to be coming from the chief cells in the stomach. Okay, that's the first way. And the second way is that this distension is going to cause a response from the medulla oblongata to produce gastrin. And gastrin is an exocrine. Okay, right over here. Gastrin is an endocrine hormone that is going to go through the bloodstream. And finally, it's going to arrive and it's going to do some things. And a part of the lower part of the stomach is going to start producing HCl and pepsinogens. And let's just focus on pepsinogens for a second. Let's zoom in on them. So pepsinogens are enzymes. And what happens is that the pepsinogen is gonna be stored in a cell, right? And then once it gets secreted by the cell, it's gonna fall into the stomach. And the stomach is a place where it's really acidic. So the pH is around 2 to 3, which is extremely acidic. And what's going to happen here is that the pepsinogen is going to get activated by the hydrochloric acid that is in the stomach. And after that happens, it will turn into an enzyme called pepsin, which breaks down proteins into polypeptides. Okay. Now we have that out of the way. We are in the stomach and we are kind of demolishing this food by using our hydrochloric acid. So the job of HCl is to denature the proteins and to unfold them and get them really out of that tertiary folded up state. And we're going to get them back into normal polypeptides. Another job of HCl is to kill the microbes and all the pathogens that are residing in the stomach, so we can create a really healthy environment for the things, because we don't want infections and bacterial stuff, things like that. Next, we will be moving on into the, large intestine, small intestine. Here, we're over here right now. So in the small intestine, we can break it down into the duodenum, the jejunum, and the ileum. So we're going to start out by zooming in on the duodenum. And over here, what's going to be happening is that, let's just recap over here. We started out with breaking down the proteins with pepsinogen. And over here, we broke down the starch with our salivary amylase. Now, what's going to be happening is that we're going to focus on our lipids, and we're going to also zoom in on the amylase, on the starch that we never finished digesting. Because in the mouth, the food doesn't stay for a long time, very short period of time spent in the mouth. So it doesn't really have time to be broken down by all the amylase in the mouth. That's why when we zoom into the small intestine over here, small I, we're going to say the duod first. When we zoom into the duodenum, we're going to see that it also has a partner. Its partner in crime would be, where is she? Hello? Guys, I don't see okay, anyways, we have what did I do? Okay, we have a pancreas in here. It's around here. Oh. Okay, so this little thing is the pancreas. And what's going to happen is that the pancreas is going to secrete pancreatic juices. So the pancreatic juices consist of enzymes and other things. Some of the enzymes that are in it are, put pancreatic in front of any of these things. Pancreatic. We have lipase, which breaks down lipids. We have amylase, which breaks down starch. We have trypsin, which breaks down proteins. And we have, there's one more. I'll tell you, peptidases. Okay. Peptidases? Yeah, something like that. These are breaking down proteins. So what is going to happen now is that the pancreas is going to throw these juices into the small intestine via something we call the pancreatic duct. Duct basically just means that it's like a tunnel. It's like a pathway where the things that are produced in the pancreas are going to have a way, they're going to find a way to go from here down to here without messing up too many things and getting mixed up. So the duodenum is where the pancreatic juices are going to be dumped. And that is why we're going to have the lipids are going to be broken down. Oh, but before this step, since we're talking about lipids, it is very important that we also need to mention that over here from the liver and from the gallbladder, which they didn't label, gallbladder, we are also gonna, let's change colors. We are also gonna have bile. So we have bile in here. Bile is produced in the liver, stored in the gallbladder, and it's also gonna be thrown into the small intestine, into the duodenum. So first, lipids are gonna be emulsified. Emulsified means they're just gonna be kind of broken into smaller pieces. So this is your big lipid. It's gonna be broken into lipid droplets. We call them droplets, which are smaller. So once they're emulsified, the emulsified lipid droplets are going to be broken down by lipase, by pancreatic lipase into fatty acids and glycerol. Okay. Now the amylase is going to break down the starch into its monomers and its diastomers, and its dimers. I don't know, something with two, into its like disaccharides. That's what I mean. So we finished our lipase, we finished our amylase. The peptidases are going to break the proteins into amino acids. Trypsin is also going to break proteins into amino acids. And then finally, we get to the ileum. So these monomers are going to start flowing down to the ileum. And they're finally going to get here. Now, what is happening? What happens in the ileum? Well, I'll tell you. We have ileum. Let's get my handwriting. It's getting progressively worse. In ileum, we have absorption. So this is when all the nutrients that we broke down are going to finally be put to use. So in the ileum, we have these structures. Yes, guys, we're talking about villi. Called villi. And they are there to increase the surface area to volume ratio. And on these villi, we have we're going to talk about microvilli. We have little microvilli. And the importance of these microvilli is to increase the surface area to volume ratio even more, so we can get the maximum nutrient absorption possible. Now, if we zoom in onto this micro, you can, guys, pay attention to the colors I'm using here. This is a microvilli. Oh, no, I can't alongside it. It's a microvillus. We have some special things. So, first of all, we need to say that our little microvillus is made out of, one layer of cells. So each one of these is a little cell, and it has one layer of cells to shorten diffusion pathways. That's exactly what it sounds like. No need for explanation. Just to make diffusion easier, for things to go in and to go out, they have to pass through the cell membrane, and that is way easier when there's only one cell. And we want it to be easiest because we need all these nutrients. Now, also, we have something called a lacteal. 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 Anyways, this is a kind of sort of vessel that leads to the lymphatic system. Now, what this is its importance is that it's responsible for taking lipids. So remember, L for lacteal, L for lipids. So that's what these are doing here. And last of all, I already used red. Lastly, we're going to have our little I don't know if these are really good. Anyways, these are our capillaries. Capillaries are the smallest blood vessel, and their importance is that they're going to take the nutrients, the really small monomers, and they're really small and thin, you know, the ideal blood vessel. And they're going to take these nutrients and they're going to transport them into the blood. And this is going to be everything else other than the lipids, not the L for lipids and the lipids are going in the lacteals. Everything else is going into the blood. Okay, now let's take this one step further and really look into those one cell microvilli structures. So what we're gonna have here is we're gonna have lots of proteins and we're gonna form really tight junctions so we avoid leakage because we don't want to leak any nutrients anywhere. We really want to keep them in control. So we're going to create really tight junctions. Now, we also need to have a lot of Golgis. So the Golgi bodies are there to package and process proteins, and we need that for transport. We're also going to have a lot of mitochondria. So we're going to have a lot of mito because we need a lot of ATP to do some diffusion, to facilitate our active diffusion. And things like that. And yeah. Also, we're going to have a lot of vesicles. So we need vesicles to, throw things out, throw things in without getting damaged. It's like a fragile warning. You can just throw things everywhere. They'll have a little packaging, so you'll be good. And yeah, that is about it for digestion. What can I say, guys? It's a very active document. Anyways, if you have any questions, feel free to ask me. And as always, thank you for watching. See you again. Bye.